Hi guys, Miss Francis here. Today I'd like to talk to you about the fluidity of membranes. We learned that the fluid mosaic model is accepted by scientists as the current arrangement of the phospholipids and proteins in a plasma membrane. Well, what does that fluid part mean? Basically, membrane molecules are held in place by relatively weak hydrophobic interactions, which enables the lipids and some of the proteins to draft laterally from side to side. Rarely would they flip-flop. But they more commonly will drift side to side. So if there's any disturbance in the plasma membrane is able to go back to its original shape. And these lateral movements are relatively fast, about 2 microns per second. And again, many of the larger membrane proteins move, but much slower. So how could scientists actually determine how fast the membrane proteins were drifting? Well, two scientists basically labeled proteins of a mouse cell with one color and proteins of a human cell a different color. They enabled those cells to fuse, forming a hybrid cell, and in less than an hour they noticed that the two colors were intermixed, and that's how they determined how fast or in this case relatively slow the membrane proteins move. There are factors that would affect the fluidity of the membrane. For example, temperature. As temperatures cool, my phospholipid tails are more closely packed, so the membrane becomes more solid. Furthermore, it depends on the type of fatty acid that's in the phospholipid tail. For example, membranes that are rich in unsaturated fats are more fluid because that kinked arrangement prevents tight packing. On the other hand, where you've got saturated hydrocarbon tails in my phospholipid, you'll notice that my plasma membrane is more tightly packed. This is where cholesterol is going to play a role. Cholesterol is a steroid, and it will be wedged between phospholipid molecules in animal cells, so that in warm temperatures, it restrains the movement and reduces the fluidity. Otherwise, it would be too fluid. And in cool temperatures, it prevents that tight packing. It prevents the tails from coming too close to one another. So here are 3 factors that enhance membrane fluidity and 3 factors that decrease membrane fluidity. So as far as enhancing goes, if I have more unsaturated fatty acid tails, if I increase the temperature, and in animals, if I increase the amount of cholesterol at lower temperatures. If I wanted to decrease the fluidity, I would add more saturated fatty acids in the tails. I would decrease the temperature. And in animals, if I increase the cholesterol at higher temperatures, the cholesterol will actually restrain that phospholipid. So whether or not I have cholesterol really depends on the temperature that is that the plasma membrane is being exposed to. That's where I'm going to end it today. Next time we'll talk about the mosaic property of cell membranes.